Okay, let's talk about viruses. They are these ultimate agents of change, right? Constantly evolving, constantly reshaping our world. In this explainer, we are going to unpack the evolutionary playbook they use, the one that lets them generate this staggering diversity, jump between species, and yeah, sometimes trigger a global pandemic. All right, let's dive in.
For decades now, we've been in this constant rolling battle. It goes way back. Think about the emergence of AIDS in the 80s, a virus that totally redefined our understanding of infectious disease. Then you had the H1N1 swine flu in 2009 that put the entire world on high alert. And this isn't ancient history, right? This is the world we live in.
And then, of course, the coronaviruses showed up in a big way. First, there was SARS, back in 2002. That was really our first major pandemic scare of this century. It was a pretty brutal wake-up call. Then about a decade later, MERS, Middle East Respiratory Syndrome, popped up, proving that this viral family was a really persistent and deadly threat.
But the threats just kept coming, and from totally different viral families, too. The Ebola outbreaks in West Africa. They showed us just terrifying lethality and also the incredible courage of the healthcare workers on the front lines. And all the while, the question of avian influenza, or bird flu, was just kind of looming in the background. Was it quietly cooking up the next human pandemic?
The pace never slowed down for a second. The Zika virus epidemic came along and revealed this devastating new danger to unborn children. And then, well, the COVID-19 pandemic, which literally brought the entire world to a standstill. I think the message from the last 40 years is crystal clear. Viral emergence is a relentless, defining challenge of our time.
So how do we actually make sense of all this chaos? As honors-level microbiologists, we need to get past the headlines and really dig into the fundamental science. So by the end of this, you should be able to describe and compare the key evolutionary processes that create virus diversity, explain how that diversity lets a virus infect certain hosts, and really understand the massive real-world impact all this has on disease emergence.
Okay, so to even begin, we have to grasp the sheer scale of the challenge. We aren't just dealing with a few dozen known bad guys here. We are living in a world that is just saturated with viruses. And that's the whole concept of viridodiversity.
Which, of course, brings us to the big question. We know about the viruses that have already jumped to humans, sure. But what about all the ones we haven't found yet? Just how big is that viral reservoir that's hiding out in nature?
Well, based on some amazing research by Anthony and his colleagues, if we just extrapolate from a single type of mammal, we can estimate there are, at a bare minimum, 320,000 undiscovered viruses in mammals alone. Just let that sink in for a second. The viruses that caused every single pandemic we just looked at, they are just the tiny, tiny tip of a colossal, invisible iceberg.
So, where does all this mind-boggling diversity come from? It's not a static thing. It's actively being generated by these powerful and absolutely relentless evolutionary engines. So now we're going to break down the core molecular mechanisms that viruses use to constantly change and adapt.
Okay, so there are three main ways viruses generate diversity, and you really need to know these inside and out. They are mutation, reassortment, and recombination. And even though they all create genetic novelty, they work in fundamentally different ways.
Right, let's start with the most basic one, mutation. Simply put, it's an error. It's a typo that gets made when the virus copies its genome. This can happen to literally any virus, but the rate it happens is hugely influenced by things like the fidelity, or you could say the accuracy, of its polymerase enzyme. RNA viruses, for example, are famous for having these really sloppy, low-fidelity polymerases, which makes them incredibly prone to mutation.
Now, not all mutations are created equal, right? A lot of them are actually deleterious. They're harmful to the virus, and they get weeded out by selection really fast. Others are neutral. They don't have any immediate effect. But some are beneficial. Maybe they help the virus dodge an immune response, or resist an antiviral drug, or even infect a brand new type of host cell. Can you think of a specific example for each one of those?
Next up, we have reassortment. Now, the absolute key thing to remember here is that this process only happens in viruses with segmented genomes. So think of their genetic blueprint as being stored on a bunch of different mini chromosomes instead of one long string. When two different strains of a segmented virus infect the same cell, their gene segments can get all mixed and matched, or shuffled, into totally new combinations in the next generation of viruses. And influenza A is the absolute textbook example of this. Its genome is split into 8 separate RNA segments. And what's really critical is that the genes for its main surface proteins, that's hemagglutinin, the HA, and neuraminidase, the NA, are on different segments. So what that means is an avian flu and a human flu could co-infect, say, a pig cell, shuffle their segments around, and boom, you could get a completely new virus with maybe an avian HA and human internal genes. This is the classic recipe for a pandemic.
Okay, and finally we have recombination. Now, unlike that wholesale segment swapping we just talked about with reassortment, recombination happens within a gene or a genome. It usually happens when the viral polymerase starts copying one genome, then physically jumps ship and continues copying on a second different genome that's inside the same cell. The result isn't a simple shuffle, it's a true hybrid, a mosaic genome made of parts from both of its parents.
So let's put these two side by side because it is so important that you get the difference, okay? Recombination creates mosaic genomes. It does this by exchanging parts of genes through template switching. This is a huge deal for viruses like coronaviruses and HIV. Reassortment, on the other hand, creates chimeric genomes. It does this by swapping entire gene segments when new viruses are being packaged, and this is the specialty of influenza viruses. Understanding that distinction is absolutely fundamental.
Okay, so we've covered the molecular nuts and bolts. Let's connect these processes back to the real world. How do these tiny little evolutionary events scale up to cause massive, widespread disease and, global disruption?
Let's look at a really clear example of mutation driving the emergence of a new disease. For pretty much all of history, dogs were not natural hosts for influenza A. But then, around 2004, an equine H3N8 flu virus picked up just a few key mutations, and those mutations were enough to let it jump from horses into dogs, where it established a completely new circulating canine influenza. A few simple typos in the genetic code opened up an entire new species for that virus to go after.
And the consequences of these evolutionary jumps, well, they can be staggering. Have a look at this graph. What's really interesting here is the overall trend. For the entire 20th century, you see this almost continuous rise in life expectancy in the US. Better sanitation, medicine, nutrition. It's a story of steady progress. But look really closely, right around 1918.
You see that? That sharp, cliff-like drop? That is the impact of a single virus. The 1918 Spanish flu, a novel H1N1 virus that we believe emerged through reassortment, was so devastating that in 1 year, it single-handedly wiped out more than a decade of human progress. That is the tangible, brutal power of viral evolution written in human lives.
So all this brings us to our conclusion. And this is not a story with a neat little ending, because it's not over. It's an ongoing evolutionary arms race between us and the viral world. And those engines of viral evolution, they never, ever stop running. So the crucial point to take away from all this is that the combination of constant, small-scale mutation and these dramatic large-scale shifts you get from reassortment and recombination, it creates this relentless engine of novelty. And this engine is continuously testing the boundaries between species, it's probing for weaknesses, and it's always generating new viral threats.
And that leaves us with a final and pretty sobering thought. We know the mechanisms, we have seen the devastating consequences, and we know that there are hundreds of thousands of undiscovered viruses circulating in wildlife right now. So, given all that, the question for our generation of scientists isn't if the next pandemic is coming. The real questions are, what will the virus be? Where will it come from? And will we be ready when it finally does?